So welcome back in another video lecture of molecular biology. So today I have brought a very important subtopic from lactose operon. So today we are going to discuss about how in presence of glucose lactose operon slows down. Okay, that is our today's topic. So this topic is commonly known as catabolic repression or the effect of glucose on lactose. First of all, let's discuss how it was started. So as we have discussed in our previous video that Jacob and Monod were the two developers of operon concept, right? So during their discovery, they find out that when glucose and lactose are both present in the media, bacteria will prefer glucose first, then lactose. Okay, and as a result we will have a biphasic growth curve or dioxic growth curve. Okay, biphasic means there are two log phase, as you can see. Okay, so the question that was in their mind, that why despite presence of lactose operon, why bacteria prefers glucose first rather than lactose? Okay, is there any mechanism by which lactose operon is blocked at that time when glucose is present in the media, or there is any kind of molecular mechanism that simply slows down lactose operon? That is why glucose consumption by bacteria is in high amount rather than lactose consumption at that point. So what is the thing going on here on the molecular biology level? So that is the question that was going on in their mind at that time. So two questions that we need to answer: why glucose is consumed first and lactose second, and when glucose is present in large amount in the medium lactose operon runs very slowly or stop completely. Okay, so I am for now just providing the answer, that is yes, it runs very slowly. So why and how glucose show its effect on lactose operon, that is our today's topics of discussion. That is how glucose stops very high activity of lactose operon, or how glucose slows down the lactose operon's activity. That is our today's topic of discussion. Okay, and this topic is famously known as catabolic repression. So let's discuss. So there are various things that I have written, but I am going to discuss with you it one by one. So the first of all, before going to know about glucose effect on lactose operon, you need to know about cyclic AMP. What that thing cyclic AMP is. So remember, cyclic AMP is a secondary messenger molecule, okay, that will simply provides or simply sends a signal that in the cell there is a scarcity of ATP. Okay, that means ATP is in basal level, there is no too much amount of ATP. Okay, that is the concept. Okay, so how it is going on? Suppose due to some intense work, cells ATP is slows down rapidly. Okay, when the ATP level is in basal level, okay, at that time the remaining ATP will be converted into cyclic AMP with the help of an enzyme known as adenylate cyclase. Okay, and this cyclic AMP will act as a secondary messenger and sends a signal to cell that you are in ATP deficient condition. Okay, so that is the concept regarding cyclic AMP. Okay, so sorry for the interruption. So now remember one thing that I going to discuss with you before I am going to discuss about the main phenomenon going on here, that the concentration of cyclic operon, cyclic AMP is inversely proportional with the concentration of glucose in the cell. Okay, that means what? That means whenever cyclic AMP is present in high amount in cell, in normal cases, make sure that in that cell the glucose amount is lower. Okay, why? The answer is very simple. There is a deficiency in glucose, means there is a deficiency in ATP. That means the remaining ATP is converted into cyclic AMP for sending the signal to cell that there is a ATP scarcity. Okay, so remember one thing, that when glucose concentration in the cell is lower, the cyclic AMP concentration will be higher. That is the concept. Okay, now let's move to the next slide. So here we are going to discuss about how cyclic AMP works. So this cyclic AMP will react with another compound called catabolic activator protein or CAP and result in a formation of Capcam complex. Okay, now this Capcam complex will bind with a specific overlapping region that is just before the promoter of the structural gene called Capcam region. Okay, this is a overlapping gene that Jacob and Monod first missed. Okay, but then they find out this region. So what is the utility of Capcam complex? We need to know. So remember one thing, Capcam complex simply enhances the transcription rate by 250 times by helping RNA polymerase that going to bind in the promoter with the help of sigma factor. Okay, so that means remember one thing, the binding of RNA polymerase in the promoter is not very of high affinity. Okay, that means RNA polymerase possess a very lower affinity to bind with promoter in reality. Okay, but the presence of Capcam complex in case of lack of operon promotes the binding of RNA polymerase with the promoter sequence. Okay, now let's move to the next slide again. So this is the thing, that Capcam complex will bind, and as Capcam complex will bind, RNA polymerase will start its transcription. Okay, so that's the normal case. So as I have told you that when glucose amount is less, there will be high concentration of CAMP, right? But when glucose is present in the environment or environment surrounding the cell, what will going on? Glucose concentration is higher, that means automatically CAMP concentration will be lower. That means what? There is very low amount of CAMP complex formation, right? So there will be low amount of RNA polymerase promoter binding, and as RNA polymerase cannot bind, and if you have watched already the negative regulation video, so there will be, I think that you know that there will be a induced repressor molecule also there. That is the product from what? That is the product from LACI gene. So due to the two side hindrance, RNA polymerase cannot start its transcription. Okay, I hope the concept is now clear to you. So that is the funda how glucose controls the lactose operon, and this concept is commonly known as catabolic repression. The concept is very simple. Whenever in the surrounding environment glucose concentration is higher, the cyclic AMP concentration will be lower, and as cyclic AMP concentration is lower, there will be very low amount of Capcam complex formation. Okay, and as there is a very low amount of Capcam complex formation, so no RNA polymerase or very small amount of RNA polymerase, that I have told you at the beginning of that class, that the operon will run very slowly in presence of glucose. Okay, so very small amount of Capcam complex will bind with that overlapping region and promote the RNA polymerase running. Okay, so as a result, the operon will run at a very slow speed. Okay, at a very negligible rate, or you can say. But when glucose is not present, okay, there will be high amount of camp formation. This camp will going to bind with cap, and now this Capcam complex will bind with that overlapping region and promotes RNA polymerase to bind with promoter. Okay, and so that, and as there are no repressor molecule present in the operator because it is inactivated with the help of allolactose, so now RNA polymerase can easily run and starts its transcription. Okay, that is how negative regulation and catabolic repression together play a important role to stop lactose operon when there is no need of lactose operon. Okay, so that is for today's class. So I hope that this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. So another thing, that this is a concept diagram, that when glucose concentration will be higher, Capcam complex will be lower, RNA polymerase cannot bind efficiently, operon will be repressed. Okay, similarly, when glucose concentration is lower, that means there is too low amount of ATP, so ATP with the help of adenylase cyclase converted into CMP, and CMP will complex with CAP, and now this CMP complex concentration is higher, and as a result it can bind with the overlapping region and promote RNA polymerase binding, and the operon will open. Okay, so that is also a concept diagram. So now I will end up that video here. So thank you for listening to this class. Take care.